All right. Well, welcome everybody. It's my pleasure to welcome you all to the ASBMB Lipid Research Division seminar series. I know many of you are used to attending these on Wednesdays, so I appreciate your flexibility to join with us today. We're really lucky because we have two great seminars today, really at the intersection between lipid metabolism and mitochondria. So we're going to hear from Zach Baker, who's currently at Washington University, but you'll hear is in transition, and then Sarah Wong from the University of Utah. And so what's great is both the speakers today are basically on the verge or have started their independent research groups, and it's really exciting to hear all the awesome science that's going on really at the interface between lipid metabolism and organelle biology. So with that, I'd like to introduce our first speaker today, Zach Baker. So Zach did his PhD with Scott Leary at the University of Saskatchewan before moving on to do his postdoc with Dave Pagliarini at Washington University. He's been just amazingly successful in Dave's lab. So he has this beautiful story that was published in Nature Cell Biology on how he used multiomics to characterize how tag lipases are used to drive kind of recovery from mitochondrial stress. And then today in this seminar, he's going to be talking about another lipid-related mitochondrial story, really characterizing the interplay between phosphatidylinositol regulation and coenzyme Q biosynthesis. And excitingly, just learned that Zach's going to be starting his position May 1st, so coming up very soon at the University of Saskatchewan, where he did his PhD. So with that, I'll stop and let Zach have the floor to share his cool work.
All right. Sharing here. All right. Okay, so thank you, Jonathan, for that introduction. Again, for those of you who don't know me, my name is Zach Baker. I am a postdoctoral fellow in the Pagliarini lab at WashU, and I'm really excited today to talk to you about this news story, some unpublished data that we have. It's currently in revision, and it centers upon one of our favorite biomolecules in the Pagliarini lab, and that is coenzyme Q. So what is coenzyme Q? Well, coenzyme Q is a redox-active lipid. I have the structure here. Consists of this long hydrophobic isoprenoid tail attached to a redox-active head group. Now, because of this redox-active head group, it allows CoQ to both accept and donate electrons, and the cell uses CoQ as a cofactor for many different redox reactions. We most often think of CoQ in its canonical role in the electron transport chain, where it accepts electrons from complex 1 and complex 2 and donates them to complex 3 to facilitate the flow of protons across the intermitochondrial membrane and establish the proton motive force that's eventually harnessed by ATP synthase to generate the bulk of cellular ATP. However, CoQ has many other roles within the cell. It's critical for nucleic acid biosynthesis, amino acid metabolism, ferroptosis, and beta oxidation. And CoQ is so important that it's actually spawned a massive industry to sell CoQ to people. So I'm sure you've all seen these in your local Costco or Walgreens. But the problem with these supplements is that they don't really work very well. That is because of this long hydrophobic tail that means that CoQ is very poorly absorbed by the body, and it's very difficult to get supplemented CoQ to where it needs to be active. This is not just a problem for people buying these supplements, but it's a critical issue for the treatment of patients with CoQ deficiency. There are many ways that you can get CoQ deficiency. We often think of it in terms of primary CoQ deficiency. This occurs when you have a pathogenic mutation in a gene that encodes for a member of the CoQ biosynthetic machinery. These have very diverse symptoms, these diseases, and they affect almost every major organ system. And while primary CoQ deficiency is quite rare, what's much more common is CoQ deficiency driven by secondary causes. These are diseases that where we see CoQ deficiency, such as neurodegenerative diseases, cardiovascular disease, diabetes, pretty much any disease that affects mitochondrial function will result in CoQ deficiency. So we have a condition in CoQ deficiency that's much more common than people realize, and we're unable to treat it with supplementation. One other potential avenue for treatment might be to boost CoQ biosynthesis at the source and just make more of it within the cell. However, to do that, we need to have a better understanding of how CoQ is made, but also how that process is regulated. So how is CoQ made? Well, CoQ is made in the mitochondria from precursors which are imported from the cytosol. So the tail, which is made from IPP, is imported and then stitched together and then attached to the head group, which is derived from 4-HB. This makes the critical intermediate PPHB or polyprenyl hydroxybenzoic acid. This intermediate is then modified by a group of enzymes that we call complex Q, which form a metabolon in the cell and act on the head group of PPHB to give it its redox-active properties and make mature CoQ. So at the onset of this project, as I mentioned, I was very interested in finding genes or proteins that regulate this biosynthetic process. And so to start that, I mined an old data set in the lab. This was our lab's Y3K data set. It was published just about a decade ago now, where we looked at over 170 individual yeast knockout strains, grew them up, and then did proteomics, metabolomics, and lipidomics, measuring thousands of biomolecules in every individual yeast strain. And so I began by asking a simple question of how CoQ changed across all of these individual yeast knockout strains. So over here, we have strains that lose CoQ, and predictably, we see these CoQ biosynthetic enzymes named CoQ and then a number. But I was really interested in the genes over here, where we knock them out and we see an increase in CoQ abundance. And one of these genes that popped out to me was this uncharacterized protein FMP30. So FMP stands for found in the mitochondrial proteome. And at the start of this project, that was really all we knew about this enzyme. So what jumped out to me was not only was it mitochondria and it was uncharacterized, but it had a predicted enzymatic function in KEGG, and importantly, it had a mammalian homolog. So I set out to functionally characterize FMP30 and determine its role in CoQ metabolism. So we generated a fresh knockout, and as you can see in the FMP30 knockout, we have higher levels of CoQ. It's about twofold compared to the wild type. And interestingly, this increase in CoQ correspondingly has a decrease in this intermediate PPHB, which results in an increase in this CoQ to PPHB ratio. And this is a metric that I'll be talking about using quite a bit throughout the talk. And so these results suggested to us that potentially we had an increase in flux through this biosynthetic pathway. We have more Q and less PPHB. And so to measure that, I performed heavy labeling experiments where we treated cells with heavy labeled 4-HB for a period of time, and after pulsing them, measured the amount of CoQ that was being synthesized. As expected, in the FMP30 knockout, they're making more CoQ, again suggesting that the flux through this pathway is increased. So if we then stop this pathway, so here we knock out one of these complex Q proteins and halt the pathway at PPHB, then we knock out FMP30, we no longer see this increase. And so this suggested to us that the step in this biosynthetic pathway that was the flux was increased was in this head group modification stage from PPHB to CoQ. Okay, so now we know that FMP30 knockout has increased biosynthesis, but how does the function of this protein result in that? So to begin characterizing the function of FMP30, I turn to its mammalian homolog. So as I mentioned, FMP30 has a mammalian homolog. That protein is NAPE-PLD. These proteins are highly homologous. The biggest difference is that FMP30 has a mitochondrial targeting sequence and a transmembrane domain. NAPE-PLD is not mitochondrial. However, the back part of this protein, this metallo-beta-lactamase domain, is very highly conserved between the two. These metallo-beta-lactamases, this is a very diverse, large protein family. They are hydrolases, but they have very diverse substrates. However, given the homology between the two, we can see here one of the active sites in this metallo-beta-lactamase domain, all of the active residues are fully conserved from yeast to humans. we thought that potentially FMP30 might have a similar function to NAPLD. So what is the function of NAPLD? Well, as the name suggests, is a phospholipase-D enzyme that acts on NAPes, or N-acyl phosphatidylethylamine. So as a PLD, it will hydrolyze this bond here to generate the corresponding phosphatidic acid and then the N-acyl ethanolamine. And these are potent signaling molecules in mammalian cells. So we wanted to see if FMP30 was performing this function as well. So we purified both NAPLD and FMP30. We verified their activity using just a generic substrate and then tested those enzymes with NAPes. However, while NAPLD will readily hydrolyze these NAPes and generate the corresponding NAE, FMP30 had no detectable function towards NAPes. Furthermore, when we went back to our yeast system, we can see our FMP30 knockout that has this increased CoQ to ppHb ratio. We can rescue this with wild type FMP30. However, we are unable to rescue this with either a catalytically dead FMP30 or NAPLD where we attach the mitochondrial localization sequence of FMP30. And so we really think that both of these enzymes have diverged in substrates and that FMP30 is not performing the same reaction as NAPLD, but given that its activity is required for its function in CoQ biosynthesis and its homology between FMP30 and NAPLD, we thought that potentially it might be acting on a different substrate. And so we tested our purified FMP30 with a panel of different phospholipids and then measured the abundance of phosphatidic acid, which will be a common product in these PLD reactions. And the only phospholipid where we saw activity with was phosphatidylinositol, suggesting that FMP30 was not performing this reaction here, but was instead performing a phospholipase-D reaction here, hydrolyzing PI, generating phosphatidic acid, and free inositol. So this is in vitro, but what about in vivo? Well, if we go back to our yeast and we isolate highly purified mitochondria from our FMP30 knockout cells, and then do lipidomics on that, each of these dots is an individual lipid species. And we've colored the PIs in blue, and you can appreciate that every species of PI in these highly purified mitochondria is increased when we knock out FMP30. If we do the inverse, we overexpress FMP30. This is going to increase that hydrolase function within mitochondria. We get the inverse phenotype, where we're now losing PIs within the organelle. So how does we really believe that this is the function of FMP30, that it's hydrolyzing PI and regulating PI within the inner mitochondrial membrane. But how does that relate to an increase in CoQ flux through the CoQ biosynthetic pathway? Well, as I showed before, we know that the step that's increased here is this conversion from ppHb to CoQ, which is performed by this group of enzymes called complex Q. So potentially, we thought that maybe the abundance of this complex is increased in the FMP30 knockout. So we performed proteomics and we looked at the FMP30 knockout. We found that this was indeed the case, that all members of complex Q were increased in the FMP30 knockout. And while this was only a slight increase, if we go back and look at that Y3K data set and look at how these proteins are changed across all of those individual yeast strains, we find that FMP30 has the second highest increase out of all 170 plus individual yeast knockouts. So there's something about the regulation of PI that is increasing the abundance of this complex Q. Furthermore, we know that complex Q exists spatially within mitochondria. So this is a paper that we published 6 years ago in collaboration with Jordi Nunari's group, where we looked at how these complex Q, where they're localized within mitochondria, and we found that they have high localization of these metabolons within what we called CoQ domains. And so we thought that potentially the FMP30 knockout might also have differing CoQ domains. So we looked at these with microscopy. Here we're tagging CoQ9. This is one of these complex Q proteins. Our mitochondria is in blue. You can see these CoQ domains. If we then look at the FMP30 knockout, hopefully you can appreciate that we have an increase in both the number of these domains and their relative intensity. This is this is quantified here. Again, knockout of FMP30 increases CoQ domains and importantly increases the partitioning of CoQ9 and these complex Q proteins within these domains where they're going to be more efficient as they can act as a metabolon. So we think this is the mechanism by which CoQ biosynthesis is increased in these cells, but how is an increase in PI resulting in this? So we're still working on this, but we have some data to suggest that the complex Q is also spatially close to PI within the membrane. And to test for this, we did an immunopurification where we pulled down complex Q and then performed a lipid extraction and then did lipidomics on these extracted lipids. And what we found was that these lipids were enriched not just for CoQ as well as other Q intermediates in the biosynthetic pathway, but also for many abundant species of phosphatidylinositol. And so we have a model here where we think that in wild type conditions, FMP30 is hydrolyzing PI and keeping PI levels normal within the mitochondria. But when you lose FMP30, you increase mitochondrial PI. This increases CoQ domains and CoQ domain abundance, and that increases flux and efficiency through the biosynthetic pathway, resulting in an increase in CoQ. And this model allows us to do some predictions. Primarily, what if we alter the amount of PI within the inner mitochondrial membrane through other mechanisms? So here I have, again, this is the function of FMP30, where it hydrolyzes PI to generate phosphatidic acid. But what if we express a different enzyme? In this case, we target a PI-PLC. This is going to mitochondria. This is going to hydrolyze mitochondrial PI and generate, rather than phosphatidic acid, diacylglycerol. Well, when we do this in both the wild type and the FMP30 knockout cells, we're able to rescue this CoQ to PPHB ratio in the FMP30 knockout. Again, suggesting that PI is the bioactive molecule that's really driving this CoQ phenotype. Now, what about if we do the reverse? Now we're targeting an enzyme that's going to make more PI within the mitochondria. This is phosphatidylinositol synthase. When we do that, we get the inverse phenotype, where we increase the CoQ to PPHB ratio in both the wild type and the FMP30 knockout. So that's the abundance of CoQ, but what about our domains? Again, we have our wild type cells here. When we overexpress FMP30, driving that PI down, we decrease our number of domains per cell. We see the same phenotype when we overexpress PI-PLC. And then when we overexpress PIS1, increasing the amount of PI within the cell, now almost the entirety of our complex Q is localized spatially within these domains, increasing, again, that biosynthetic flux. So this is our model for how CoQ is increased under FMP30, but can we use this under more physiological context? And so to look at that, we wanted to see how FMP30 knockout affected CoQ patient mutations in yeast. And so I mentioned before that there are many identified cases of primary CoQ deficiency. We focused on patient mutations in CoQ7. This is one of these complex Q proteins. And we generated the corresponding mutations in yeast and expressed these. And when we do that, we lower CoQ levels, as you might expect. These proteins are not working very efficiently. But when we knock out FMP30 on top of that, we can partially rescue the CoQ levels in both of these, in yeast expressing both of these patient mutations. And similarly, if we look at respiratory growth, so we're growing these yeast in conditions that require mitochondrial function and CoQ levels for growth. They're deficient. These mutants are deficient in this growth. But if we knock out FMP30 on top of that, we can partially rescue this respiratory growth, and including this mutation, which was completely respiratory dead, but we've now allowed it to grow a little bit when we knock out FMP30. So this is all in yeast, but everyone wants to know, what about in a mammalian cell? Now, I mentioned before that the mammalian homolog for FMP30 does not do the same function, and it's not mitochondrial. And so to try and interrogate this question, we instead used our mitochondrial localized PI-PLC model, where we expressed this and we transduced it into U2OS cells to drive down the levels of mitochondrial PI. When we do that, we see a similar phenotype as we saw in yeast, where reduction of mitochondrial PI reduces the amount of mitochondrial CoQ, and similarly, it results in a significant growth defect when we grow the cells in media that requires mitochondrial function. In this case, it's galactose-containing media. Now, we were really interested in this because this growth defect was much more significant than we would expect based on just this decrease in CoQ levels, which suggested to us that potentially other mitochondrial functions might be impaired by the loss of mitochondrial PI. When we did proteomics on these cells, we saw that there wasn't much of a decrease in any of the proteins that are involved in oxidative phosphorylation. And so we instead wanted to measure how OXPHOS complex formation. We did this by complexome profiling. For those unfamiliar with complexome profiling, it works on the same process as Blue Native Page. So we isolate mitochondria, and then we run those mitochondria. We separate those complexes using Blue Native Page. Rather than in a traditional Blue Native page where you might transfer that for a Western blot, we instead cut this gel into many different slices, and then we prep all those slices for proteomics. And now, thanks to modern advancements in mass spec technology, we can perform many different mass spec experiments and measure the protein, all detectable proteins in each of these individual slices. And so rather than just blotting for an individual complex, we can look at the abundance of all of these individual subunits of the different OXPHOS complexes. So we can see here in a wild type cell, this is as we go down the gel, and we look at complex 1 in particular, it's primarily within these higher molecular weight supercomplexes in the wild type. But if we look at our cells overexpressing mitochondrial PIPLC, we lose this abundance in these super complexes, and instead we have higher abundance in these lower molecular weight subcomplexes. And so while the actual abundance of these proteins is not changing, we believe that PI may potentially be important for the formation of these complexes. And so in conclusion, we've shown that FMP30 is a phosphatidylinositol phospholipase-D enzyme. It's localized to the inner mitochondrial membrane, that the regulation of PI levels by FMP30 controls the efficiency of CoQ biosynthesis. We believe that PI is important for not just complex Q formation, but also for other mitochondrial complexes. And this is really exciting because PI is a critically understudied lipid. Despite the fact that it represents actually a pretty significant portion of the mitochondrial lipidome. So in yeast, it represents about 15%, which is even higher than the levels of cardiolipin. In mammalian mitochondria, it's a bit lower. It's about 5 to 8%, depending on the measurements. But you often never hear about PI and what its roles might be in mitochondria. And many reviews that talk about mitochondrial lipids don't even mention PI, or if they do, they talk about it in the context of phosphatidylinosyl phosphates. But we think that PI may have many different roles. And excitingly, too, we also know that PI is dynamically regulated under conditions of mitochondrial dysfunction. So this is in yeast when we inhibit mitochondrial dysfunction and have respiratory dysfunction, we increase the abundance of major PI species, and we also increase the expression of PI synthase. And so we think that potentially this might be a way for the cell when it's undergoing respiratory dysfunction to increase mitochondrial PI and stabilize some of these mitochondrial complexes, such as complex Q. So with that, I'd like to thank the lab and particularly my supervisor, Dave Pegorini, who has been a terrific mentor, as well as the other co-first author on this paper, Rachel Guerra. And then, as Jonathan mentioned, in May, I'll be starting my own lab back at the University of Saskatchewan, which is coming up very soon. And so with that, I will take any questions. Thank you.
Cool. Thank you for an amazing talk, Zach. It was beautiful work. I have a quick question for you. I also wanted to tell everyone to use the raise your hand feature, and then can call on you if you have any questions. But I'll get us started. One of the questions I had was, I know that it was shown those CoQ foci are spatially kind of linked to the ER, and I'm curious in your FMP30 knockout if you get more incidences of ER mitochondrial contacts, and if you think there's interplay going on there.
Yeah, that's that what we hoped that was going to be the case. We don't see these domains. So the domains have been linked to ER mitocontact sites. We see it is a smaller percentage of them that are, most of them aren't, spatially around these mito-ER contact sites. And we also, when we knock out FMP30, we don't really see a change in the number of mito-ER contact sites, but that is a model that we've thought a lot about and that we're interested in pursuing in the future because it could be really interesting if PI might have a role then as well in increasing or altering those mito-ER contact sites. There has been other papers that have shown that kind of larger scale papers that have shown that knockout of FMP30 can alter mitochondrial morphology. We did not see that in our yeast when we looked at it, but again, that might also speak to alterations in ER mitocontact sites that might be affecting division and mitochondrial fusion.
Cool. All right. We have questions from, I'll first call on Anam and allow him to talk.
Hi, Tay. That was a super talk. I have a very specific, possibly technical question. I think I
Mhm.
saw on your slide that the PIPLC was targeted to the inner mitochondrial membrane,
Mhm.
or I'm not sure what I read, but does it matter where it is, considering that the lipids are possibly exchangeable across leaflets and between membranes?
Yeah, so we targeted it using the FMP30, both the FMP30 targeting sequence and the transmembrane domain, and then we kind of attached the PI-PLC on top of that. We did not look if that would if we targeted it somewhere else, if that would also similarly affect it. I mean, even potentially targeting it on the other side of the inner membrane. We're not sure exactly how PI is transported from, presumably it's made either at the ER or at the outer mitochondrial membrane and how it's transported into the inner membrane. So I can't say whether or not if we put it elsewhere, if that would affect the phenotype. But if I had to guess, I would say that if we reduce total cellular PI, we would probably see similar things because that PI is going to be flowing into mitochondria somehow.
You.
Okay, we have a question from Thomas Ballo.
Can you hear me?
Yes. Yes, I can.
Yeah, great talk, Zach. Congratulations. You may be aware of the fact that our lab was one of the first to show PI on the surface of mitochondria.
Hmm.
Recently, Josh Pemberton in our lab showed this, so this is really exciting for us. My question is there a mammalian homologue of this? Because you said the one is not doing the same job and it is even not in the mitochondria. So
Hmm.
is there a similar enzyme, or is it possible that the lipid is different for the mammalian regulation?
Yes. Yeah, terrific question. And yeah, thank you for a lot of the work on PI. Like this PI PLC was we got the idea from one of your lab's papers. Um, we think that for the mammalian homologue, yeah, again, NAPLD is not functioning in the same way. There are uncharacterized beta lactamases within the inner membrane, mitochondrial intermembrane space that we are looking into to see if they have a similar function. That's one of the future directions of this project. We have not yet identified a mammalian homologue. It is possible that, and probably likely that some of these functions of PI are going to be different from mammals to yeast. But we do think that it will have some kind of role and so I'm particularly invested in trying to find this mammalian homologue and testing some of these uncharacterized beta lactamases, some of which have been linked to lipid metabolism before, and seeing if they have any function, any activity against PI.
Thanks.
Okay, I think we have time for one more quick question.
Oh, hi. Beautiful talk, Zach. I have a question. We study lipid metabolism and de novo lipogenesis and mitochondria in the ocular system. I have a question whether CoQ defects are directly rescued by lipid pathway intermediates.
Do you mean pathway intermediates in the
Mm-hmm.
biosynthetic pathway?
Yeah.
Yeah, so we can. I don't know if I can go back here to. The short answer is yes.
Mhm.
And so if we go back, depending on there are intermediates within this pathway here that we
Mhm.
can rescue. We can bypass some of these individual, if we have a mutation in one of these genes, we can bypass that by providing head groups that will already have that modification. But one thing we run into is that similarly to CoQ, as soon as we attach this tail, it
Mhm.
makes it very hard to deliver. And so it's difficult to bypass or rescue this by feeding the cells these specific intermediates. And so we're kind of limited to only adding in the head groups. And there have been groups that have looked at supplementing with 4-HB that can boost CoQ levels in some circumstances, or potentially bypassing, again, as I said, some of these mutations with different head groups that already have those modifications.
Okay. Thank you so much.
Well, thank you, Zach, for a phenomenal talk. In the interest of time, I'll ask that people maybe forward their questions to the chat box, and then and Zach can answer them there. So I'll go ahead and introduce our second speaker of the day. So we're going to hear from Sarah Wong, who did her PhD with Lois Weissman at the University of Michigan studying organelle transport. And then she joined Adam Hughes's lab for a postdoc at the University of Utah, where she's been studying this fascinating stress-derived compartment on the mitochondrial outer membrane. And basically is involved in the removal of parts of the outer membrane. And so she spent her postdoc working on understanding the role of lipid saturation in this outer membrane quality control process. Sarah is now a SRI fellow at the University of Utah since last July. And this is a program focusing on the mentorship of undergrads in research. And in that position, she's continuing in her group to study aspects of lipid saturation on cellular behavior. So with that, I want to make sure that Sarah has as much time as possible, and I'll go ahead and let her get going.
Great, thank you, Jonathan. Let me get this set up. Okay. Right, yeah, thanks for having me. I'm Sarah. I'm currently at the SRI, but today I'm going to talk mostly about the work that I did as a postdoc with Adam Hughes, and then touch a little bit upon the work that I'm doing now as an independent fellow. And so the Hughes Lab is, hold on, I'm having trouble advancing. Sorry about that. Okay, so the Hughes Lab is really interested in understanding how a breakdown in cellular organization leads to aging and disease. And at the core of cellular organization is organelles. And so the Hughes Lab has been focusing on mitochondria, which are important for energy production, amino acid metabolism, and cellular signaling. Because these organelles are so crucial for cellular function, there's a wide variety of quality control mechanisms that make sure that mitochondria function and maintain homeostasis even under stress and disease. And so here I'm showing a few different pathways that handle excess proteins in mitochondria. And the Hughes Lab focuses on the mitochondrial-derived compartment pathway, which I'll call MDC for short. And MDCs were first discovered in yeast, but have since been described in mammalian cells and C. elegans. So based on research over the past 10 years, we now know a wide variety of stressors can induce MDC formation. This includes metabolic stress, such as high intracellular amino acids or impaired TCA cycle, changes in membrane composition, including changes in lipids such as phosphatidylethynolamine or PE, as well as changes in lipid saturation, which is what I'll focus on today. And then we also see MDC formation under protein stress. And this includes protein overload due to mistargeted or surplus proteins on the outer mitochondrial membrane. MDCs themselves form at mitochondrial contact sites. They contain multiple lipid bilayers of only outer mitochondrial membrane, and they also sequester specific cargo proteins. These structures can capture cytosol, and they can be removed from the main mitochondrial tubule through fission machinery. MDCs can be sent to the vacuole, which is the yeast equivalent of the mammalian lysosome, via autophagy, but this is mitophagy independent. And so overall, we believe that MDCs may be mediating mitochondrial metabolism and regulating the outer membrane composition, but there's still a lot of open questions as to how they're regulated and what their roles are in the cell. So here I'm going to show an MDC forming in a live yeast cell. If you've never seen the mitochondrial network in yeast, it's really dynamic. Here we have TOM70 tagged with GFP. TOM70 is an outer mitochondrial membrane protein and known MDC cargo. And in our wild type untreated cells, you can see that this the mitochondrial network is extremely dynamic. Now, when we add an MDC inducer, in this case rapamycin, you now see TOM70 forming this bright puncta that is then elongated into this tubule. And this tubule will eventually snap back onto itself to create this large luminal compartment. And so here it's more of a sphere. And we believe MDCs are forming through this series of tubulation and forming back onto itself and forming these multilamellar structures. And so this video on the right is going through different slices of EM. It's going to color in pretty soon, and you'll see two different outer membrane lipid bilayers, here in blue and here in green. And then the main mitochondrial tubule is in purple and the ER is in yellow. And so we believe that this tubulation event occurs, it forms and folds back into a sphere, capturing cytosol, and then this can occur multiple times to create multiple lipid bilayers or multilamellar structures. And because these structures are so rich in membranes, it really begs the question as to how lipids might be regulating this type of remodeling. So the first study that found that lipids might have a role in the MDC pathway came from a previous graduate student, Tinyao Zhao. And she found that low levels of phosphatidylinositol can promote MDC formation. On the other hand, low levels of cardiolipin can actually inhibit MDC formation. And so her study really focused on different phospholipid species, which are defined by their head groups. But we hypothesized that other membrane properties of lipids would impact MDC biogenesis. These are extremely membrane-rich domains. There's got to be other things that are regulating this pathway. And so we wanted to focus on the fatty acid tails or the saturation state of phospholipids. And to do this, we focused on OLE1, which is the only desaturase in yeast. It's equivalent to mammalian SCD1, which stands for sterile-CoA desaturase. And OLE1 takes saturated fatty acids and adds a double bond to create an unsaturated fatty acid. OLE1 itself is increased in expression in tight lipid packing conditions, including low oxygen levels or low temperatures. And these unsaturated fatty acids, because they have this now kinked tail, the kinked chain, when they're incorporated into lipids, can push these molecules apart, and it creates a looser packed lipid membrane. Often people will say more unsaturated lipids will increase membrane fluidity, but membrane fluidity is also affected by other factors such as proteins in the membrane and other lipid species as well. Here is a proof of concept experiment showing that if we overexpress OLE1, we can increase unsaturated lipids in the cell. In these blue dots are phospholipid species where both of those fatty acid tails are unsaturated, and these species are vastly upregulated. On the other hand, phospholipids where one or both tails are saturated are downregulated. And so this means that we can use this mutation and ask what happens when there are high unsaturated fatty acids. The punchline is that it induces MDC formation, but I'm going to walk us through this microscopy experiment first. So in our empty vector control cells that are untreated, here we just have DMSO or vehicle control, you see both TOM70 and TIM50 nicely localized to the main mitochondrial tubule. Again, TOM70 is our outer membrane protein and known MDC cargo, whereas TIM50 is an inner membrane protein that is not incorporated into MDCs. If we treat cells with vapamycin, we now see formation of MDCs, and so we see this bright TOM70 positive structure that is also negative for TIM50. And this is how we quantify MDCs. Interestingly, when we overexpress OLE1, we now see constitutive MDC formation. So even without treatment, we see MDCs forming in these cells. And then when we add an MDC-inducing condition on top of this, such as vapamycin treatment, we now further increase the percentage of cells that form MDCs. And so this data was really exciting. It tells us that increasing unsaturated fatty acid synthesis leads to constitutive MDC formation and may promote further biogenesis of MDCs. But it doesn't tell us if MDCs are forming because we have high unsaturation or because now there's an imbalance between saturation and unsaturation. And so I've looked at high saturated lipids in a variety of different ways. This includes using strains from Ite Boudin's lab, which downregulate OLE1 expression by using different promoters, as well as looking at SCT1, which is an acyltransferase that preferentially incorporates saturated fatty acids into phospholipids. But here I'm going to show you our experiments with MGA2, which is a transcription factor that promotes the expression of OLE1. And so again, in our wild-type cells, untreated, we don't see MDCs forming. If we add an MDC inducer, in this case, concatamicin A, we now see MDC formation. And interestingly, when we delete MGA2, we don't see any constitutive MDC formation in these untreated cells. So this tells us that high saturated lipids is not sufficient to promote MDC formation. And in fact, when we add an MDC-inducing condition, such as concatamicin A, we can inhibit MDC formation. So this overall tells us that MDC formation might be promoted by high unsaturated lipids, and that high saturated lipids can actually inhibit MDC biogenesis. MGA2 again is a transcription factor and it has many targets. And so we can really nicely rescue MDC formation if we overexpress OLE1 in our MGA2 deletion mutant. And so again, high unsaturation leading to MDC formation. Overexpression of OLE1 itself can promote both membrane expansion and lipid droplet synthesis. And so to figure out which side of the coin is responsible for MDC formation, we turn to PA1, which is equivalent to mammalian lipin. And when we delete this regulator, we can push fatty acids into lipid droplet synthesis into storage. And in this situation, we can actually inhibit MDC formation. And I'm not showing you today, but for the sake of time, but if you shunt fatty acids into membranes and into membrane biogenesis through phosphatidic acid, you can promote MDC formation. And so together, this is telling us that high unsaturated fatty acid synthesis, specifically high unsaturation in membrane lipids, is likely driving MDC biogenesis. And because it's driving it through the membrane, we really need to go back and ask what is happening to the outer membrane composition that might be forcing or promoting MDCs to form. So previous work in the lab, this is from a previous postdoc, Zach Wilson, he found that MDCs may clear excess or surplus proteins. And so in this experiment, we're looking at the TOM complex. The TOM complex is a large multi-subunit complex on the outer mitochondrial membrane, and it facilitates import of proteins into the outer membrane. In wild-type cells that are treated with vapamycin to induce MDCs, we don't see TOM22 or any other TOM complex subunit localizing in MDCs or co-localizing with TOM70 here. However, if we delete TOM6, this mutation disrupts the TOM complex and prevents proper assembly. And in this context, we now see TOM22 as well as other TOM complex subunits co-localizing with TOM70 in MDCs. And so this study really found that if you disrupt the Tom complex, you can now get these surplus proteins and subunits in MDCs, presumably to clear them. Interestingly, we see similar phenotypes when we overexpress OLE1. So again, in our empty vector control cells, Tom complex subunits do not localize in MDCs. If we delete Tom6 and disrupt the Tom complex, we now see Tom22 and other Tom complex subunits in MDCs. And we see this as well when we overexpress OLE1. And so this microscopy experiment is really nice. It's great to see these subunits in the MDC, but it doesn't quite answer if we have an intact Tom complex in these MDCs, similar to a Tom6 deletion. And so we used blue native page analysis to look at if Tom complex is intact or not. And so with blue native page analysis, we isolate mitochondria and run them on a non-denaturing gel. In our empty vector control cells, you'll see the migration of this large molecular weight band, and this band indicates intact Tom complex. However, when we delete Tom6, we now see the migration of a lower molecular weight band, and that indicates that the Tom complex is disrupted. And we see a similar banding pattern with OLE1 overexpression, telling us that high unsaturated lipids might be disrupting the Tom complex, and perhaps these excess proteins are being cleared by MDCs. And so the summary for this project is that we found increasing saturated lipids may inhibit MDC formation, whereas increasing unsaturated lipids promotes MDC formation, and this may be through outer membrane protein stress. It still remains to be seen if there are potentially lipid metabolism effects that are also contributing to this phenotype. There's some other open questions related to MDCs and lipids. We don't know if there's any specific biophysical properties that are driving MDC formation. I think it would be really cool to figure out if high membrane fluidity is necessary and sufficient to form MDCs. We also don't know if MDCs sequester specific excess lipid species. So we know that MDCs can sequester specific protein cargoes, but do they also sense and sequester and remove excess lipids? And which lipids are these? And then it's still unclear what role MDCs play in mediating lipid stress. We know that high unsaturated fatty acids can be promoting MDC formation, but are MDCs a response to this, and is this part of cell survival or cell health under this type of lipid stress? And so these questions are all really interesting, and I'm excited to figure more out about MDCs. But in my new position at the Science Research Initiative, I'm also interested more broadly in understanding how excess unsaturated phospholipids affect membranes and membrane proteins. And so in my position right now, I'm working with a group of 17 undergraduates and leading an independent research program asking a wide variety of questions. And so to kick off our research program, we began with a GFP screen. We took the yeast GFP collection, where the majority of yeast proteins are tagged with GFP. We incorporated our mutation of interest, OLE1 overexpression, where now we have high unsaturated fatty acids. And we did high throughput microscopy in this 96-well plate format to identify proteins that are potentially mislocalized in high unsaturated fatty acids. I'm showing you one example of a potential candidate here. So this protein is normally localized to the nuclear periphery, but in high unsaturated fatty acids, it now localizes in these puncta. And so the vast majority of students in the lab are currently following up on this screen. They're tagging proteins with GFP, and they're making mutations to alter lipid saturation. And so hopefully these studies will tell us a little bit more about how unsaturated fatty acids are impacting organelle composition, protein function, organelle function, and general cellular homeostasis. There's also a couple other projects in the lab. So overexpression of OLE1 can confer resistance to certain potential harmful chemicals, and also increases glycerol production, which is related to osmotic stress response. And so some folks in the lab are trying to understand the mechanisms behind how high unsaturated fatty acids might position cells to resist these types of stresses. High unsaturated lipids can be both detrimental in some contexts, but also beneficial in other contexts. And so we're trying to figure out in which context you might actually want high unsaturated fatty acids. We're also interested in polyunsaturated fatty acids. So Saccharomyces cerevisiae only makes monounsaturated fatty acids through OLE1, and any polyunsaturated fatty acids come from the media, which actually really positions cerevisiae to be a good, clean model to study the roles of unsaturated fatty acids. And so we are now incorporating polyunsaturated fatty acids into cerevisiae and trying to figure out the impacts of these lipids on organelles and stress response pathways. And so with that, I'd like to thank the Hughes Lab and all the folks involved in that project, as well as past and present members. The metabolomics core here at the University of Utah, who did a ton of lipidomics for this project and the funding that supported a lot of it. And then I'd like to thank my current home at the Science Research Initiative, the directors and program manager, and then all of the students in the lab, as well as Marcus Babs and the imaging core for use of their microscopes. And I'm happy to answer any questions.
Well, thank you, Sarah, so much for an awesome talk. I'll just remind everyone that if you raise your hand, you can get your questions, but I'll start us off again. So one of the things I noticed in your imaging was that when you overexpressed OLE1 and looked at TOM70, you could see kind of constitutive MDC formation, but also what looked like a lot of small puncta distributed throughout the cytosol. And I'm kind of curious if the saturation state could be influencing mitochondrial fission, or also maybe indirectly if it could be influencing autophagy and the breakdown of these compartments.
Yeah, that's a great catch and a really good question. Overexpression of OLE1 is pretty detrimental to cells. They grow a little bit slower. It's actually lethal if you work with it in a haploid, or not quite lethal. It's pretty detrimental. It is possible that other things are going on here as well, which is part of the reason that I decided to do a GFP screen and look more broadly. There's a previous study that did find that increasing unsaturation can actually promote formation of the pre-autophagosomal structures. So it could definitely be affecting other quality control pathways involved. I never really looked specifically to see if these MDCs end up in the vacuole. I haven't done GFP clipping assays, but yeah, that would definitely be a good question to ask for sure. Thank you.
All right, we have several hands raised, so we'll go ahead and start letting people talk. So I'll call on Anat Manan first.
Sorry, Jonathan, I guess it is me that you called on because I'm able to unmute myself. So Sarah, I have a question for you. Arguably, POR1 is the most abundant protein in the outer membrane. Is that captured in the MDCs as efficiently or as TOM70 or not at all?
No, we don't see POR1 in MDCs. We're still trying to figure out, and the Hughes Lab is actively working on trying to figure out how it's decided which proteins get into MDCs or not. Zach Wilson's study about mistargeted proteins identified that proteins mistargeted from the ER, so he was looking at what happens when you disrupt the GET pathway. And so those proteins that can't go through the GET pathway can be mistargeted to MDCs, as well as disrupted complexes like the TOM complex. There's some, and so we're still trying to figure out all the signals involved. It's been bounced around that maybe ubiquitination might be involved in tagging proteins and getting them into MDCs. But yeah, there's still a lot of open questions for that. But no, POR1 is not in MDCs.
Thank you.
Okay, we have a question from Daniel Raven.
It was a very nice talk. I just have a quick question. The data you have on unsaturated versus saturated fatty acids is really interesting. I think it's probably very important, but it begs the question about the asymmetry across leaflets. And I'm wondering if you've thought about targeting a scramblase to either the outer membrane or the inner membrane, or in some way disrupting the asymmetry of the leaflets.
Yeah, that's a fantastic question. Yes, for a while we were trying to figure out how to get inner versus outer lipidomics, or maybe like whole mitochondria versus inner mitochondria only lipidomics. That's a fantastic question, and that's also ongoing research. So there is a graduate student in Adam's lab right now who's thinking about the mucose complex, which connects the inner membrane and the outer membrane. And we do have a working hypothesis that MDCs might be forming in response to the inner membrane as well, where maybe MDCs might be removing excess outer membrane based on the needs of the whole mitochondrial balance, which is affected by the inner membrane. A scramblase is a great idea. We haven't done those experiments, but yeah, I wouldn't be surprised if you're correct. There's imbalances between saturation and unsaturation, but also between inner and outer membrane that are leading to this quality control pathway.
That's interesting. Very good. Thanks.
Thank you.
Okay, we have a question from Nancy Hart.
Yes, I think it's probably more of a comment than a question. Over the last century in the United States, we have undertaken a vast experiment where our dietary lipids are about 6 times what they were 100 years ago. And we also know that these dietary lipids are incorporated into mitochondrial membranes within just a few hours of consumption. So I think this is just an absolutely beautiful, fascinating research, and I can't wait to jump online and find out more about what your lab is doing. So thank you.
Thanks, Nancy. Yeah, that's a great comment. And it is really interesting. So we did treat yeast with oleic acid, which is the product of ole-1. And interestingly, putting oleate in the media, even if you have 220 to like solubilize it and emulsify it and get it into cells, it didn't trigger massive MDC formation. And I think it's because cells are pretty good at mediating this type of stress. And so it took a really harsh mutation to get this quality control pathway, because it's probably having so much stress to deal with for so long. I am still really interested in trying to understand the difference in response to dietary unsaturated fatty acids versus genetic mutations. And I think this would be really interesting for health and disease mechanisms as well, because you're right, like you can get dietary issues, but also SCD1, which is the mammalian equivalent of ole-1, is shown to be upregulated in metabolic disease, cardiovascular disease, cancer, neurodegenerative diseases. And so I do think that cells subject to the stress due to genetic changes are probably responding in a way where maybe there is a threshold of dietary lipids that are going to do something similar. So thank you.
If we look at it, though, in terms of modulating membrane viscosity, the oleate is probably going to have less effect than the polyunsaturates and the long-chain saturates. So it might be interesting to look at oleate as the stressor, rather, or not oleate, but polyunsaturates rather than oleate as the stressor. Thank you.
Yeah, thank you.
Okay, we have a question in a chat box that I can read to you. So it's from George Carmen, who asked, says, one of the effects of the PA1 deletion is an increase in unsaturation of PA. And in our paper showing that, we question whether ole-1 was involved. So do you think that the effects you see in the PA1 deletion were due to increased unsaturation as well as the increase in phospholipid synthesis?
Oh, yeah, that's a good question. So we deleted ICE2. We had trouble actually deleting PA1 itself. And I have not done lipidomics with these mutants. But no, that's a really good point. I think it's hard to tease apart everything going on. Sometimes I do feel like I'm playing lipid whack-a-mole, like I'm, increasing this and it's going to decrease something else in compensation. But I wouldn't be surprised if there's probably some feedback loop of like, okay, you aren't making this type of lipid and it's affecting the overall membrane properties in a certain way. And so now you have a compensatory change in a different membrane property. I hope that answers that question.
So I guess we have a minute left. I wanted to ask one question that I still wonder, which is, and maybe can be more speculation because I don't think you have the answer to this, but do you think at the end of the day, it's the lipid composition of the outer membrane that's driving the ability of these MDCs to form? Or do you think it's more that you're disrupting mitochondrial processes and you're getting unassembled protein complexes and that's driving the MDC formation?
Yeah, that's a really good. Yes, I think about this a lot and I have not figured it out yet. I think I think you need to have a membrane that is physically able to form MDCs, right? And maybe that's why we're seeing this inhibition with high saturation. I do have a biophysics student in the lab who's very interested in modeling, and he's really interested in trying to model how changes in membrane composition might allow this type of structure to form. We've also tossed around the idea of taking like isolated mitochondria and using optical tweezers, and it's like if we just pull it, can it now form this type of structure? In terms of, I think the unanswered questions for that is like we don't know enough about the inherent membrane properties that might be required for this. And then we also don't know enough about the physical machinery that's involved. So we don't know if there's a motor or an adapter or, ER regulated thing that is physically pulling these tubules out to make this MDC. Sorry, I have so many slides. Make this initial structure before it forms an MDC. And then also once you get this structure, how do you snap it back into this large sphere? Like I always have trouble kind of visualizing these, like the intermediates between these intermediates. So I guess yes, I am also speculating about this. And I think it'll be really interesting once we figure it out.
Cool. Well, thank you, Sarah, and also for Zach for like phenomenal talks. It's great to have you both, and I'm really excited to see kind of the science that's going to continue out of your lab. So thank you again for making it for a great seminar today.